Okay, everybody, we are going to finish up this Unit 2. You guys will have a test on Thursday. So let me get to where we left off. We left off at proteins. Okay, so proteins are important because they're about 50%, and we may have already covered this, but we are going to cover it again. Just to kind of pick up where we left off. Proteins make up a huge portion of our body. They're about 50% of our body weight. We need proteins because they help us in the form of enzymes, structure. You guys think about, mostly you think about protein as being muscle, but it's also used as collagen and elastin that we basically lose over time the ability to regenerate those things. For muscle contraction, actin and myosin are some of the most important proteins or amino acids or proteins that we have in our body. They're for transport, so like our blood transportation, protein transportation, so that we can have RNA and DNA, and then hormones such as insulin. Also hormones such as thyroid hormone, pituitary hormones from the pituitary gland. So we have to have proteins basically for our body to function. Those four main elements of the body: proteins, carbohydrates, lipids. Those are and RNA and DNA. Without if we're missing any of those, we will be in big trouble. Most.
I need all seniors to report to the cafeteria, please. All seniors report to the cafeteria. Thank you.
Sorry, guys. So proteins contain carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus, and those are also essential minerals that our body needs that we must consume. So when we think about the kind of diet we have and consuming things, our body can't make its own proteins. We have 13 essential amino acids that our body can't make itself. We can't make our own lipids, our own carbohydrates, or our own. I mean, we have our DNA, but we must have those and RNA. But we have to have those other nutrients so that our body can replicate cells that have worn out or replicate life. So the amino acid structure. You have one nitrogen and two hydrogen atoms that make up an amino group. They're generally called, you also have a carboxyl group. That would be a carbon, two oxygens, and a hydrogen. So the first one is, on the right side is that carboxyl acid group. On the left side, it has that nitrogen group. And they are the amino group, and those are bonded together by carbon, always. Then you have the R group, which is designated for 20 different, they designated a R group, and they are 20 different amino acid groups. They have specific properties based on which R group they are, whether it's a peptide or a peptide bond, whether it's an amino acid. So basically, we get that peptide bond is created from an amino acid group, like a carboxyl group joining with an amino group, and through dehydration reaction, where it pulls out the water, then you get that peptide bond and you get a dipeptide or two peptides. So just remember that when you are, like you guys should have learned in chemistry, if you have like chemical AB, like that amino group, and you have CD, through dehydration you can pull out the H2O, and then you can have your yielded compound. But if it's not a dehydrated reaction, then you're going to have, if you have A and B and C and D, say those are your bonds or your groups, then when they yield something, it may be AC and BD, but things can't just come into a compound that weren't already there in those reactive groups. Peptide bonds link together amino acids to make polypeptides, which are a whole protein. So there are 4 levels of organization. You guys will need to know your 4 levels of organization. There's the primary structure, and that is basically a chain of polypeptide chain. It's a bunch of amino acids, a specific sequence of them being together. And the secondary structure would be a coiling or folding chain. So as you can see in that second group, they're there with a hydrogen bond. And then there's those amino acids or links. So you have an alpha helix and a plated sheet that come together to make that secondary structure. The tertiary structure is a polypeptide bond, but it's a single subunit that it's basically what we would think of as when we think of what a molecule would look like. It's a whole three-dimensional shape of the polypeptide, and it's created when the R groups bond. So the primary is just a sequence or a chain of amino acids to make a chain polypeptide. Then the secondary, it's a folding or coiling kind of chain. The tertiary looks like a three-dimensional, kind of like we would think of what a whole structure would look like. And it's created when R groups bond. And then the quaternary structure it's two or more polypeptide chains together. So it can have 2, 3, 4 chains together to make that structure. Amino acids are assembled into polypeptide chains according to what they're going to be doing. So we may need a quaternary structure for DNA for a specific kind of coding, but we may need you can need any of those. And so when we think about RNA and how important protein is to RNA, it's just as important to DNA and for coding structures, or for coding so that we can replicate cells. Now think about it this way: if you don't have the right protein structure, or if you don't have the right links in the chain, then your DNA will not replicate appropriately, which can cause all kinds of genetic issues. On the cellular level, all the way through to your health. So chemical reactions and enzymes. Now, our body cannot function without enzymes, so that chemical reactions can happen, and we can transform things that we ingest or things that we come in contact with. We have to have those enzymes to break them down so they can be used. A chemical reaction is a process that changes or transforms one set of chemicals into another. You did a whole lot of that, I hope, in chemistry with stoichiometry and other things like that, where you had a product and it yielded a different product. Chemicals that enter into the reactions are the reactant, and when what is made is the product. So if you have A and B plus C and D, those are the reactants, and the product would be what it yielded: A and D and B and C. Those chemicals. It changes the bonding pattern to yield a specific product. Energy is released or it is absorbed when chemical bonds are formed or broken. So we have to have the right enzymes for things in the body to happen. If we think about it through, like in the digestion process, we have amylase in the mouth. Amylase is an enzyme that helps break down starches. And it starts those chemical reactions, breaking down starches as soon as we put them in our mouth so that they can be used. We can grab that glucose and break it down into ATP quickly. We have peptide, or we have amylase. We have peptide. Now that my brain is going blank, let me just go back and start over because we'll talk about that in a minute. So when we release energy, that happens spontaneously. Sometimes when it's absorbed, that's when energy starts from them. Some energy is released during an activation, or sometimes energy is needed to actually break down products. So when we think, and we do most of that in our body through digestion, but it also happens when insulin is released. It happens with reactions in the brain when we have stimulus related to cold or hot or any kind of reaction our body has. We have to have some kind of enzyme to make those chemicals break down and for us to be able to utilize them. So enzymes that are vital proteins that run biochemical reactions. So if we look at the reaction chain on the right, you see the enzyme is available in an empty active site. So when we take something in, like a simple sugar that has sucrose in it, then that substance it sits down in a specific enzyme react or sets down in a specific reaction. And what happens is, just like it shows, the specific enzyme that's needed to react has a open link. And so whenever we chew up food and it starts to break down, it'll have a specific um, substance that will sit right in that reaction, that enzyme, to activate it. Then lots of times it'll pull water out. Once that amylase starts to work, or it could be lactase, depending on whether it's sucrase, which is what this would be for sucrose. It's sucrase. So it sits down in there. It starts that reaction. It starts to break down that sucrose into fructose and glucose. And then when those are broken down, then that enzyme is available again for the next time it needs to break something down. The reaction, they work based on substrates. Those substrates are specific pieces that will sit down in there. Most enzymes have an ACE behind them, like sucrose has sucrase, lactase breaks down lactose, amylase is a liver enzyme, but it also helps break things down. All enzymes, like I said, are very shape specific. They're like a lock and key. So you have to have the right key to unlock that enzyme to make it work. And usually one enzyme equals one substrate. So that means one enzyme will create one reaction to break down one specific thing. Enzymes have a unique three-dimensional shape so that they can fit into their specific substrate. So that would be only sucrose can fit into sucrase. Sucrose can't fit into lactase. It doesn't have the right lock for that, the right key for that lock. Shapes determine the function in which chemical reactions are performed, and they all kind of they all have a specific pattern on how they work over time, and then go back to their dormant stage until they're needed again. Factors that affect enzyme activity: temperature, salt concentration, pH balance. Back when we talked about pH at the beginning of this, I told you how important pH is, and it truly is. Your pH balance cannot get off, or enzymes can't react. And if that happens, then basically body function shuts down at the organ and then at the cellular level. Enzymes, they are inhibitors that can alter. There are certain inhibitors, enzyme inhibitors can alter enzyme functions. So a competitive inhibitor can block an active site or a substrate, and a substrate can't get in and make that activity happen. An example of that is when we have pain, our brain has these enzymes that are looking for our own neurotransmitters to help us stop having pain. So we have this active enzyme. When we take a pain pill, even if our body, we're still getting that reaction that like when you break a bone, you have pain for a long time after that. But initially they give you pain medication. What the pain medication does is it goes and it sits inside those enzymes so that reaction letting your brain know you're hurting can't activate. So it's sort of like a little Pac-Man that has the little piece of cheese cut out. That pain medicine fits right in there for 4 to 6 hours, depending on what you're taking, and then it's all dissolved. It's all gone. That the medication in your body has the enzyme or that enzyme reactor spot has utilized all of that in there. And so if you're still hurting, it can activate that pain receptor. So when you hurt, that's why they tell you to take your pain medicine ever so often, because it's only available for so long inside that enzyme site that would make you know you're having pain. So everything is kind of lock and key. It has a habit. So sometimes we take things. People who don't produce enough insulin, diabetics, have the same issue. They're especially type 2. Their beta cells, they can't activate. So we give them medication that makes those beta cells active. So there are different things that can inhibit what's going on. Non-competitive inhibitors, they alter the enzyme's function by changing its shape. So say say we had an excessive amount of sucrose in our mouth, then there may be we have some kind of genetic disorder where our enzymes don't develop the right way. The body can actually change the side of that enzyme so that it can get its appropriate what it needs, make that enzyme function appropriately for the body. Poisons and pesticides, and drugs, like I talked about with the pain medication. Those are things that can definitely alter our enzyme activity and inhibit them from working. One of the things that we give people every day that inhibits function and inhibits the function of our clotting time, we give people Coumadin, which is very small doses of Purina rat poison. So it's a pesticide that we actually give people because people who have clotting disorders, have valves, have artificial things implanted, it keeps clots from building up on those. So sometimes we actually want that to happen. So think about this way. When we think about why do we put lemon juice on apples? What happens when you put lemon juice on apple? Do you know what happens, Caitlin? So if you put lemon juice on an apple, the apple doesn't turn brown. If you cut it in half and open it up, or you slice it up, you sprinkle a little lemon juice on there, or there used to be a powder you could put on them. I don't know if they still have that fruit something. But what happens is that's an acid that sits on there, and it prevents the apple from reacting with the air. So it inhibits the enzyme process because it's too acidic. Now let me ask you this. Lemon juice is about a 2 on the pH scale. So the pH scale is 0 to 14. So lemon juice is about a 2, and apple juice is about a 4. Do you know what that means? The difference in what that means? So the further down, the lower the number, the more acidic something is. The higher, the more alkaline it is. So that means apple juice has some acid in it, but it's not nearly as strong as that lemon juice, right? So you have to think about that too. Like an apple isn't really acidic. It's not. We don't think about it being really acidic, but we can put a stronger acid on it to keep it from oxidizing. Why do we have fever?
That's something to make your body move around, I think.
So if I'm sick and I have a bacteria invading my body and it needs to replicate, or a virus, and with a fever with a bacterial infection, it kind of runs a little lower. When we have a virus, it runs really high. The reason why is the temperature, your body temperature inhibits virus or bacterial growth. And our body knows, hey, this is a virus that's using my own cells to replicate, so I need a really high temperature so certain enzymes and certain cellular activity doesn't happen, and that virus can't keep replicating, and over time it'll kill it off, hopefully. With the bacteria, we do the same thing. When our temperature rises high to a certain point, that bacteria can't continue to grow because its cellular components can't survive in a higher body temperature. So what's happening to that egg when it hits a hot frying pan? It immediately starts to cook, right? What happens to, like, we know that eggs can carry salmonella. So when it hits that heat, it's basically the same thing that's happening that would happen to your body. It's killing off that bacteria. And we put perishable things in the refrigerator because we want, one, if you put it in the right bag and then put it in the refrigerator, it doesn't get exterior chemicals quick, right? If you set certain things like a tomato, if you just set a tomato out on the shelf, after about a week, it's going to get all moldy and gross. It can last longer in the fridge because it's a cooler temperature where they ripen more. Vegetables ripen and fruit ripen in heat, where in the refrigerator it slows that ripening process. Did you know you can do the same thing but leave if you take foil and wrap foil around bananas? Did you know it will slow down the top of the bananas? Do you know it'll slow down the process of them going bad? Because the banana is inside the peel, right? But the top of it is where it's been cut from the tree. And so when you cover that top part, it slows down its ability to continue to ripen. If you want them to ripen faster, then you can basically trim off a little bit of that, the stem of them, a little bit every day, and it'll make them ripen faster. Or put them in a brown paper bag and set them up on where it's warmer up high. It'll make it ripen faster. So genetic sequences, DNA, and we know that DNA, we know what that is, and we know that it has to mature at a certain rate so that cells can reproduce. The DNA, basically, it makes up who we are and how we continue to live. It's a specific sequence of proteins. It's a double helix of proteins. So if something happens and say we don't get the right proteins, because when you talked about cells before in biology, I hope you remember that you have to have all three types of RNA for cellular replication. You have to have messenger RNA, transport RNA, and then receiver RNA. And then that kind of activates all the rest of the activity in the cell, telling the cell, hey, I'm wearing out. But if you don't have all the right proteins, whether it's for RNA or DNA, then basically cells just die off and they don't replicate. Or if you have too much, or your body is overactivating that cellular regeneration, what's happening? It's overacting.
Like cancer.
It can be cancer. It can also be, do you know anybody who has like eczema or psoriasis? That's another thing that's happening. The body is producing multiple layers of keratotic part of the skin, and it's basically your body thinks that it shouldn't have its own skin. It's an autoimmune response. So we have to think about how even at the enzyme level, our DNA has to, when it replicates, it has to make sure that the enzymes that are available, are supposed to be available, are there. So as we use certain enzymes, and they'll wear out. So just like anything else, when it's overused, it'll wear out. Enzymes do the same thing. Well, through our DNA and through cellular regeneration, our body can help produce new enzymes, and so that we can break down things the way we're supposed to. So sometimes there's genetic issues, like people who are lactose intolerant. That's a genetic issue. That's usually genetic. You have tons of allergies, so some of that can be genetic. That can be genetically linked to your parent, a parent, or a grandparent, or a great grandparent. And so those are things that happen through genetic sequencing and through your DNA. Is that somewhere the first person who was allergic to something, their DNA didn't replicate to manage that, and then it continued on. And then that sequencing in their DNA that made them allergic to something, it continued to be passed on. And sometimes as it goes through each generation, the gene can mutate, kind of like it's done with you, where instead of maybe your great grandmother was just allergic to nuts, for you it's grass. What else are you allergic to? Gluten.
Wheat,
Yeah. So
soy.
are you allergic to dairy? You're not allergic to dairy. But it's kind of changed into a whole nother, it's kind of become much more extreme with you. It's something that if you choose to have kids one day, it may or may not be passed on to them. So you have to, when you go to the doctor, you always want to tell, when babies are little and kids are little, you tell them what you're allergic to also, because they start to look for that. They look for, especially with gluten sensitivity and gluten intolerance, they look at that over generations, and they may even tell you from birth, we want you to give this specific type of formula because it's got pre-digested proteins that will make your child more likely later on maybe not to have some of those soy or gluten sensitivities. They won't give it a soy formula, most likely. They will tell you to use a different type of formula that's not soy. Oh, sorry, guys. Okay. And guess what? That's it. All right. So tomorrow we will review we will review for your test. Your test will be on Wednesday if you're a Monday Wednesday. It will be on Thursday if you're a Tuesday Thursday, and you'll have your review shortly. Thanks, guys.